Okay, our today's topic is genome and human genome project. So, first we will discuss what is genome. What is the definition of genome? This term genome, it was coined by it was coined by Professor Hans Winkler in 1920. He had adopted this term genome from the Greek language, in which genome means to become or to be born. So the term genome, it was coined by Professor Hans Winkler in 1920. He had adapted this term from the Greek language. In Greek, genome means to become or to be born. However, Oxford Dictionary suggests that this term genome, it is combination of two words, gene plus chromosome. Oxford Dictionary suggests that term genome has been coined by the combination of two words, gene and chromosome. So, gene and chromosome, this and this, it has been combined and the word genome had been formed, right? So, according to scientific literature, the term was first coined by Professor Hans Winkler in 1920. However, Oxford Dictionary has some different view. According to Oxford Dictionary, the term has been coined by the combination of two words, gene and chromosome, right? So, what is this genome? How can you define a genome? In sexually reproducing organisms, there are diploid cells. Sexually reproducing organisms have diploid cells in their body, as well as they have haploid germ cells. So, in the somatic cells of diploid organisms, in diploid organisms, there are somatic cells and there are sex cells. Somatic cells, they are diploid, and sex cells, they are haploid. That means sperms and gametes, sperms and ova, gametes, they are haploid, whereas somatic cells, they are diploid. In a sex cell, in a sperm or ovum, there is only one complete set of chromosomes. In a sperm and ovum, there is only one complete set of chromosomes. In case of human beings, for example, in case of human beings, in sperm there are 23 chromosomes, or in ovum there are 23 chromosomes. So these 23 chromosomes, which are present in a sperm or ovum, they represent one complete set of chromosomes, and we call them as haploid. We write it as 1n. We write it as 1n. So all the genes which are contained in this one complete set of chromosomes in a sperm or in an ovum represent as one complete genome. So all the genes contained in one complete set of chromosomes represent as haploid genome. So similarly, in somatic cells, there are two complete sets of this chromosomes, and all the genes which are contained in these two complete sets of chromosomes, they represent the diploid gene. They represent the diploid genome, right? So now how can you define a genome? So genome in sexually reproducing diploid organisms is all the genes contained in a full set of chromosomes. All the genes contained in a full set of chromosomes is known as genome. So in somatic cells, we have got a diploid genome. We have got two genomes, one N and one N. So in somatic cells, we have got a diploid genome. In our sex cells, sperms and ova, we have got haploid genome. So genome is a full set of chromosomes along with this all genes. Do you understand? So we can define a genome as all the genes contained in one full set of chromosomes. That is the one genome. So in our sex cells, we have got haploid genome, and in our somatic cells, we have got a diploid genome, right? You know, our haploid genome is formed of some 3 billion base pairs. In our haploid genome, there are some 3 billion base pairs. What are base pairs? You may be remembering adenine, thymine, cytosine, guanine. These are the base pairs. So how many base pairs are present in one complete set of chromosomes? How many base pairs are present in the DNA in one complete set of chromosomes? In haploid genome, it is 3 billion base pairs. Then what will be the number of base pairs in our diploid genome? So in our diploid genome, there will be 6 billion base pairs. 6 billion base pairs in our diploid genome, right? Now you can tell me what is a genome. So all the genes which are contained in one complete set of chromosomes represent us one haploid genome. And in a diploid cell, we have got two sets of chromosomes. All the genes which are contained in these two sets of chromosomes, they represent diploid genome, right? So our human haploid genome is formed of 3 billion base pairs. These base pairs, they make up the DNA, and genes are also present in the DNA, right? A specific sequence of these base pairs, which codes for a polypeptide or which contributes to a phenotype, is known as a gene, right? So these genes, they are made of these base pairs, and there are 3 billion base pairs in our haploid genome and 6 billion base pairs in our diploid genome. So this is the case of genome in diploid organisms. What about haploid organisms? Haploid organisms, bacteria, archaea, or sometimes viruses. What about their genome? Do they possess a genome? If we look at haploid organisms, what makes their genome? Haploid organisms. Haploid organisms like bacteria, archaea, and viruses. They are all haploid organisms. Bacteria are haploid, archaea are haploid, and viruses are haploid. So what about their genome? You know, in a bacterium, there is a prochromosome. This is the prochromosome of a bacterium. Or we also call it as genophore. So this is the main genetic material of bacterium. It is a single circular double helical DNA molecule. In addition to this prochromosome, certain bacteria also have got plasmids, small pieces of circular DNA molecules known as plasmids. So what makes the bacterial genome? All the genes which are contained in this prochromosome and plasmids together, they form the haploid bacterial genome. All the genes which are contained in a bacterial cell, in its prochromosome and its plasmids, they collectively form the haploid genome of the bacterial cell, right? Similarly, in archaea, there is again prochromosome and plasmids may be there. All the genes which are contained in prochromosome and plasmids, they form the haploid genome of the archaea. What about viruses? In some viruses, there is DNA as genetic material. Some viruses have got RNA as genetic material. So all the genes which are contained in the DNA or in the RNA of a virus constitutes its genome. Constitutes its genome, right? So all the genes which are contained in the DNA or RNA of a virus, that constitutes the viral genome. That constitutes the viral genome. There are certain cell organelles, for example, mitochondria, which also possess DNA in them. Certain cell organelles like mitochondria, they possess DNA in them. All the genes which are contained in this DNA of mitochondria forms the mitochondrial gene. Forms the mitochondrial gene. Or there are plastids. In plastids, it will form the plastidial gene, right? So we first discussed the genome in diploid organisms. Now in diploid organisms there are two sets of chromosomes. All the genes which are contained in a single full set of chromosomes, that forms the haploid genome. When there are two sets of chromosomes, that makes the genome diploid. So we have diploid genome in somatic cells and we have haploid genome in sex cells, sperms and ova. Now, when we look at the haploid organisms like bacteria, archaea, and viruses, in bacteria, the genetic material is in the form of the prochromosome, or we call it as genophore. And in addition to this prochromosome or genophore, there are small circular double helical DNA molecules known as plasmids. So all the genes which are contained in this prochromosome and in these plasmids, they together form the haploid bacterial genome. Similarly, the case is in archaea. However, in viruses, some viruses have got DNA, some viruses have got RNA as genetic material. Those viruses which have got RNA as genetic material, we call them as retroviruses, right? So all the genes which are contained in the DNA or RNA of a virus, that constitutes its haploid genome, right? Similarly, certain cell organelles like mitochondria and plastids, they also possess DNA in their inner chamber. This DNA, which is found in the matrix of mitochondria or which is found in the matrix of this plastid, all the DNA which is found in the matrix of mitochondria or matrix of plastids, along with its genes, constitutes the mitochondrial or plastidial genome, right? So that is the definition of genome. That is what we mean by genome, right? Now I hope you understand that our sex cells, they have got haploid genome, our somatic cells have got diploid genome.
Yes, sir.
And genome represents all the genes which are contained in this one full set of chromosomes. After this, we are going to discuss about human genome project. Human genome project. This is the most important question which is frequently asked in the examination. So first we must be able to define what is this human genome project. So first we will write the definition of this human genome project. This human genome project is an international international scientific research project. Research project with a primary goal of the primary goal of determining the sequence of chemical base pairs that make up the DNA, or you can write here which make up the DNA, which make up the DNA, and to map and to identify and map all the genes contained in it, and to identify and map all the genes contained in it, and all the genes contained in it from both physical and functional standpoint. Functional standpoint or functional point of view. So this is the definition of Human Genome Project. In the examination, you are asked to define what is this Human Genome Project. So you must be able to define this Human Genome Project. So Human Genome Project is an international scientific research project. So it is not confined to a single country. When the project was started, international support was sought. So for that purpose, many countries joined with USA in carrying out this project. So when many countries participated in this project, it became an international scientific research project. So it is an international scientific research project. What was the primary goal of this project? The primary goal of this project was to determine the sequence of chemical base pairs which make up the DNA. So the primary goal of Human Genome Project, the primary aim of Human Genome Project was to decipher, was to identify the sequence of base pairs in DNA, how these base pairs are present in the DNA, right? Whenever we make a diagram of DNA, we show their adenine, thymine, cytosine, guanine, cytosine, adenine, thymine. We show them randomly. But these scientists, they wanted to know the exact sequence of these base pairs. So that was the primary goal of this Human Genome Project, to know the exact sequence of base pairs in the DNA of organisms, in the DNA of human beings, and to identify and map all the genes contained in it from both physical and functional standpoint. So another important goal of this project was to identify the genes, where the genes are located in DNA, where the genes are located, where a gene is contained in DNA, and to draw the maps by which we can easily understand the location of the gene. So this DNA, it is spread in 46 chromosomes in a diploid cell. In a haploid cell, it is contained in 23 chromosomes, right? So these scientists, they wanted to know, they first wanted to identify the different genes. Which gene controls which function, right? So they wanted to identify the genes. And they had to also map, they had to also locate the position of a gene in the chromosome, on the chromosome, right? So they have to identify the genes, they have to map the genes, they have to locate the exact position of a gene on a chromosome, right? Then they also had to study the physical structure of the gene and their function, right? So they had to sequence the chemical base pairs, When the human genome was completed, then only we came to know that our haploid genome has got 3 billion base pairs in it. The sequence of these 3 billion base pairs, it is well published now. It was published in the journal Nature. We have now the sequence of base pairs in the genome by the help of this Human Genome Project. Then scientists came to know that there are some 20,500 genes in the human genome. In one complete set of chromosomes, there are some 20,500 genes. They identified which gene controls which function. They identified which gene controls which function. And they also located the position of the gene on the chromosome. Suppose this is a chromosome. Suppose this is chromosome number 5. Now they can easily say on chromosome number 5, we have here got the gene for tallness, right? On chromosome number 5, we have got here the gene for eye color, right? So they have located the genes and they have marked their position. They have marked their position on these chromosomes. So that is known as chromosome mapping, right? So they have identified the genes, they have mapped them, they have located their position on different chromosomes, and they have studied the function of the gene. They have studied the structure of the gene and the function of the gene. So this is the definition of Human Genome Project. So its primary goal was to determine the sequence of chemical base pairs which make up the DNA. Then they had another goal to identify and map the genes. And then there was another goal to study the structure of the gene and to understand its function. This is the definition of Human Genome Project. So when this Human Genome Project was started, why this Human Genome Project was started? I think this is session for today. I have a guest waiting for me. So I hope this is session for today. Thank you for being with me.